Hi everyone and welcome to Miss Estruch Biology. This video has been so highly requested. Topic 6 for AQA, homeostasis, the nervous system, is one of the hardest topics. So it's no surprise that this is the one that you all wanted next as the entire topic video. It is a long one because there's a lot of content, but it will help you so much with revision. Now, if you do need to jump ahead at any point, then have a look at the time codes and just jump straight to it. Now, I know lots of you are struggling with revision at the moment, the time pressures, the stress. So one thing I would recommend is do not spend time making revision notes because it takes so long and that part doesn't actually help improve your memory. Now, you do still need to have a set of condensed notes quite often to help you with your revision, but that's where this resource comes in. If you haven't already seen my A-level revision notes, these are for AQA and they cover the entire theory. Now, I've been teaching for over 10 years, so I've used my knowledge to include in this set of notes all of the key words. I've highlighted those, I've highlighted the key marking points, and at the end of each topic I give you a key point summary as well as links to the essay to help you to get those top marks in the essay, linking the theory to the application, the importance of. So if you do want to have a look at them, check them out and I've linked them down in the comments. But for now, get yourself a drink, get yourself a snack because this is a long one. Let's get going. We begin with one of the key definitions, and that is of the term stimulus. So a stimulus is a detectable change in the environment, and the cells that can detect these changes are called receptors. So organisms increase their chance of being able to survive if they can respond to these changes in the environment. And we look at in this topic the different mechanisms for how organisms do this. So we begin with the responses in flowering plants. And tropism is the term given to when a plant responds to its surroundings via growth. Now, tropisms can be described as either positive or negative. Positive meaning the plant grows towards the stimulus, negative meaning it grows away. And the two stimuli that you need to know are light and gravity. Now, tropisms are actually controlled by specific growth factors, and one key example is IAA. Now, IAA is a type of auxin and it can control cell elongation, meaning making the cells get longer. And this is in shoots, it elongates, but having IAA in the roots actually inhibits the growth of cells. IAA is made in the tip of both the roots and the shoots, but it can move through by diffusion to other cells. Phototropism is the term given to the tropisms where the plant is responding to light. And for shoots, the reason that it improves their survival chance if they respond to light is because light is needed for the light-dependent reactions in photosynthesis. So plants' shoots will grow and bend towards the light source, and this is positive phototropism. Now, the shoot tip cells, as we already said, those will be producing IAA, and that causes cell elongation, causing the shoots to grow upwards towards the light source. Now, the IAA diffuses to the shady side. So in the first images, we can see that the IAA is diffusing downwards away from the light source. However, in the other images, we can see we've got a unilateral light source, meaning it's coming from one side. And picture 3 shows you the light to the right-hand side. And the IAA will still diffuse to the shady side, that just happens to be on the left. And that will cause the cells on that side to elongate, and therefore the shaded side cells will be stretching and that side will be longer, and that causes the plant to end up bending towards the light source. In the roots, we have the opposite happening. The IAA will still diffuse away from a light source, but this time we said that in the roots, IAA inhibits the growth. So as a result, the top layer of cells will be growing or elongating more, and that is why the root ends up bending downwards away from the light. And this helps survival because that will help anchor the plant further into the ground and potentially reach more water sources. So that's an example of negative phototropisms. Gravitropism is the term given for the responses to gravity. And in the shoots, the IAA will diffuse from the upper side to the lower side of a shoot. So it's diffusing down towards gravity. And if a plant shoot was vertical, then that would mean the IAA would be diffusing down, causing the lower cells to elongate, and the plant would be growing upwards. Now the image we can see down here is if a shoot was on its side, and that will cause the shoot to bend upwards, and that's because the IAA diffuses down, the cells on the lower side will elongate, and that is why it's bending up. And this is against gravity, so that would be an example of a negative gravitropism. Now in the roots, IAA moves to the lower side of the roots also, and that would cause the upper side to elongate and the root will bend down towards gravity. And that would be a positive gravitropism. And that increases survival because again it helps to anchor in the plant. Now a reflex is an example of a rapid automatic response to protect animals from danger. A reflex arc is made up of 3 neurons, and that would be the sensory neuron. We'd then have the relay neuron and the motor neuron. So it's only 3 neurons. That means you're only going to have 2 synapses, and that is one of the reasons why it's such a rapid response. Now some more simple responses are taxis and kinesis, and these keep organisms within their favourable conditions within their environment. In particular, we're thinking about light, moisture and potentially chemicals as well. Taxis is one of these types of responses, and this is when an organism will move its entire body towards a favourable stimulus or away from an unfavourable stimulus. So the example we have here is an earthworm, and earthworms don't want to be in the light. That would dry them out. So if it is light, they would move their entire body away towards a darker light source. So when an organism moves towards a stimulus, this is known as positive taxis. When an organism moves away, it's described as negative taxis. So the earthworm moving away from the light would be negative phototaxis. Kinesis is slightly different. This is when an organism changes the speed of movement and the rate that it changes direction. So if an organism moves from an area where there are beneficial stimuli in the area within the harmful stimuli, its kinesis response will be to increase the rate it changes direction. And that's to increase the likelihood that it will return itself to the favourable conditions. If an organism, though, is surrounded by the negative stimuli, the rate of turning decreases to keep it moving in a relatively straight line, and this will actually increase the chances of it moving to find a new location with the favourable conditions. Receptors we talked about at the start, so these are cells which can detect a change in the environment or stimuli, and each receptor responds only to one specific stimuli, and this stimulus of a receptor can lead to an action potential or establishing a generator potential, and that is what results in an organism being able to respond. Now there are 3 receptors that you need to know: the Pacinian corpuscle, rods and cones, which are both light receptors or photoreceptors found in the eye. So if we start with the Pacinian corpuscles, these detect changes in pressure, so they're pressure receptors, and you mainly find them deep in the skin of your fingers and feet. And this is what we're seeing here. This is a cross section through your skin, and the Pacinian corpuscles are about the same level, slightly lower than your hair follicles. Now, the Pacinian corpuscle is actually a sensory neuron wrapped in lots and lots of layers of plasma membrane. and that has special channel proteins embedded within those plasma membranes. So that's what we're seeing here. We've got our sensory neuron with the nerve ending. We've got lots of connective tissue with gel in between each of these layers. Now, the membranes of the Pacinium corpuscle have stretch-mediated sodium channels. So where we said it's special channel proteins, that's what we're referring to. And what this means is when these proteins have pressure applied to them, they are stretched and deformed and it opens the channel. And in this case, it opens these sodium ion channels. So if pressure is applied to the finger, for example, the Pacinium corpuscles deep in the skin of your finger will be stretched and deformed. That will widen the sodium ion channels, and therefore sodium ions will be diffusing in, which leads to the establishment of this generator potential. So it's diffusing into the axon within the sensory neuron, and if enough sodium ions diffuse in to exceed that threshold, then you have an action potential and a response will occur. The next two receptors are both found in the human retina, and these were the photoreceptors, rods and cones. Now, rod cells are named because of their shape. They are more rod-like in shape, and they can only process images in black and white. Now, to create the generator potential, the pigment in the rod cells, which is rhodopsin, must be broken down by light energy. Now, they can detect light at very, very low intensities. And the reason for this is many rod cells connect to one sensory neuron, which we call retinal convergence. Now, although this gives the benefit that you're able to see in black and white, even if it's very dark, because all of those connecting rod cells contribute towards reaching that threshold to generate an action potential, the downside is it provides low visual acuity. And what that means is it's difficult for the brain to distinguish between the separate light sources. So you don't have very good clarity in your vision. So that's why at night time, if you do happen to wake up, you'll be able to see, but it'll only be black and white, and you won't be able to see particularly clearly, which is why you might end up bumping into things if you try and walk. Now, the other type of cell that is a photoreceptor is the cone cells. And there are three types of cone cells, and each one contains a different type of iodopsin pigment. There's one that is for red, green, and the last one blue. And they all absorb slightly different wavelengths of light. And we don't just see red, green, and blue in our vision, but depending on the proportion of red cone cells or green or blue cone cells that are detecting a stimulus, that will give the different colours, not just red, green, and blue. Now, iodopsin is only broken down if there is a high light intensity. So you can only see in colour if there is enough light present. And the reason for that is we only have one cone cell connecting to one sensory cell. And therefore there has to be a high enough light intensity to break down enough of the iodopsin pigment to be able to trigger an action potential. Now that gives the property of high visual acuity. So because only one cone cell is connected to one sensory neuron, that means the brain can distinguish where the separate source of light was detected, and therefore you have a lot more clarity and precision in your colour vision. Now, the distribution of the rods and cone cells is not equal across the retina. You actually have most of the cone cells located in this part here called the fovea. And that is where you have the highest light intensity reaching the retina, because that is where the light is focused by the lens. Now that is an advantage because the cone cells will only be able to detect the colour lights in high light intensities. And that's why you mainly find them at the fovea. The rod cells can still detect light even at low light intensities. And that is why they are found further away, where there'll be slightly less light being able to reach that part of the retina. Now, the next thing you need to know about is how your heartbeat is controlled. Cardiac muscle is myogenic, and what that means is it will contract and relax on its own accord without any nervous system input. However, the speed at which it contracts is controlled by the nervous system. And that is what you need to know about in this section of the A-level. Now, the parts that are involved in controlling the heart rate are, first of all, the sinoatrial node, or the SAN. And this is located in the right atrium. This group of cells or tissue is known as a pacemaker, and that's because they set the pace at which your heart will beat. You then have the AVN, or the atrioventricular node, which is located between the right atrium and the left ventricle. The bundle of His is what we call the tissues running down the septum. And then you have the Purkinje fibres, which branch through the ventricle walls. Now you can see it on the outside here, but it'll be branching all the way around the ventricle walls. Now, how those all work together to control the heart rate is linked to what you would have learnt in the topic 3 when you did the cardiac cycle. So when you learnt about atrial systole and atrial distole, or diastole and systole, people say it differently. So the SAN starts off this process by releasing this wave of depolarization, or electricity, and that will spread across the two atria. And when the wave of depolarization hits the muscle, it causes it to contract. So that is why the atria contract first. When that wave of depolarization reaches the AVN, that causes the AVN to release another wave of depolarization. Now, there's actually a non-conductive layer of tissue that separates the atria and ventricles, and that prevents the wave of depolarization travelling straight down to the ventricles. So instead, what happens is that wave of depolarization that was released by the AVN travels down the bundle of His and then up through the Purkinje fibres. Now, because of that, it causes a very slight delay in the time it takes for the apex of the heart, so the base of the ventricles and the ventricle walls, to contract. And that is actually an advantage because that means it gives the atria enough time to contract and force all the blood into the ventricles before the ventricles will then contract. And the ventricles would contract from the apex first and then move upwards. And that's an advantage because it's a bit like squeezing toothpaste out of your toothpaste tube. If you squeeze from the bottom upwards, you're going to get all the toothpaste out. And it's the same here. Squeezing and contracting that cardiac muscle from the bottom, then upwards, forces all of the blood out of the heart. Finally, all of the cells repolarize, and that causes the cardiac muscle to relax, and then the cycle happens again when the SAN releases another wave of depolarization. Now, the involvement of the nervous system is what triggers the rate at which the SAN will release those waves of depolarization. And the parts involved are the medulla oblongata in the brain. That is what controls the heart rate, but this is through the autonomic nervous system, meaning it's automatic and there's no conscious control. So you can't think and control your heart rate. There are two key parts, and that is the sympathetic nervous system and the parasympathetic nervous system. And that's what we're being shown here, these two key routes that the impulses can take connecting the medulla oblongata to the SAN in the heart. Now, the sympathetic nervous system, if impulses travel via the sympathetic nervous system, that will cause the SAN to release the waves of depolarization more frequently and therefore increase the heart rate. If more impulses are sent down the parasympathetic nervous system to the SAN, that will cause the SAN to release these waves of depolarization less frequently, and therefore it decreases the heart rate. One of the stimuli that will change the heart rate is pH, and the other one is blood pressure. There are different receptors that detect these different stimuli. The changes in pH are detected by the chemoreceptors, and the blood pressure changes are detected by pressure receptors, but they are both found in the same location. You find them in the walls of the aorta and also the carotid artery, and this is the next blood vessel that attaches onto the aorta. Now, your blood pH does change, and during times of high respiratory rate, so for example, maybe if you're doing lots of exercise, the blood pH will decrease. And that is because of the excess carbon dioxide from aerobic respiration or lactic acid from anaerobic respiration. And these acidic compounds have to be removed because if you have excess acid in your blood, it can denature enzymes. So this is achieved by increasing your heart rate so that the dissolved carbon dioxide in your blood can reach your lungs faster and therefore diffuse out into the alveoli and be exhaled. So that would mean you need more impulses going down the sympathetic nervous system to the SAN to increase the heart rate. Now, for blood pressure, if it's too high, it can cause damage to the walls of the artery. So it's very important to put mechanisms in place to bring down your blood pressure. And to do that, you would have more impulses sent via the parasympathetic nervous system to cause the SAN to fire less frequently and therefore decrease the heart rate. If your blood pressure is too low, though, that might result in insufficient oxygenated blood being supplied to respiring cells. So that would mean that your heart rate needs to be increased to try and counteract this. So therefore, the pressure receptors would detect this and result in more impulses being sent via the sympathetic nervous system to the SAN to cause the SAN to fire more frequently and therefore increase the heart rate. Now, if we have a look at a myelinated motor neuron structure, the key parts are, first of all, the cell body. And this cell body contains all the organelles you'd have in a typical plant cell. So, for example, we can see the nucleus. It also has proteins and neurotransmitter chemicals which are made in that part of the cell. The dendrites we can see here branching carry action potentials to surrounding cells. The axon is the conductive long fibre that is making up this whole part of the cell, and that carries nerve impulses all the way along this particular motor neuron. Schwann cells are what we call these layers wrapping around the axon, and they form the myelin sheath, which is a lipid, and therefore it insulates. It does not allow the charged ions to pass through to the axon. There are gaps though, and these gaps are called the nodes of Ranvier, and that is where they are not insulated. A resting potential is what we would have happening inside of the axon when there is no stimulus. So when a neuron is not conducting an impulse because there's no stimulus, there's no change in the environment, there is still a difference between the electrical charge inside the neuron, so inside the axon, and outside. And we call that difference the resting potential. There are more positive ions, the sodium ions and potassium ions, outside compared to inside of the axon. And therefore, the inside of the neuron is comparatively more negative. And that is why the resting potential is minus 70 millivolts. Now, you need to know how that resting potential is established in the first place after a response to a stimulus, and how it's also maintained if there is no stimulus. Now, the key way it's maintained is by a sodium-potassium active transport pump. So this is an example of co-transport, but it is also active transport because ATP is involved. And at this pump, you have 2 potassium ions being pumped into the axon, and 3 sodium ions are being actively transported out of the axon. This creates an electrochemical gradient, and therefore the potassium ions, because there's a higher concentration of them inside the cell, they will start to diffuse out of the cell. The sodium ions, there's a higher concentration outside, so they will start to diffuse into the cell. However, the membrane is more permeable to potassium ions because there are more potassium ion channels and because some of those ion channels remain permanently open. So that means more potassium ions can diffuse out than sodium ions diffusing in. And as a result, there are more positive ions outside of the cell compared to inside of the cell, and that's how we get the minus 70 millivolts. Now, if there is a stimulus, and if it's a large enough stimulus, it can generate an action potential. An action potential is when the neuron's voltage increases beyond a set point from the resting potential. And this is what generates the nervous impulse to then travel along the neuron. So an increase in voltage, also known as depolarization, is due to the neuron potential becoming more permeable to sodium ions and therefore becoming more positive. Once an action potential is generated, it moves along the axon like a Mexican wave. So let's have a look at then how this action potential is generated. A stimulus provides the energy that can cause the sodium ion voltage-gated channels in the axon membrane to open, and this will cause the sodium ions to start diffusing in. And that's what we can see here. One of those channels has now opened, and therefore sodium ions will start to diffuse into the axon. The potassium ion channel here is permanently open, but because we now have some sodium ions moving in, we start to see this increase in the voltage. Now, if the stimulus is large enough to cause enough of these sodium ion channels to open, then you'll have enough sodium ions diffusing in to cause an increase beyond the threshold potential. And if you do have an increase beyond that minus 55 millivolts, then you will always have the rest of this curve happening. You'll always have an action potential generated. Now, there are even more voltage-gated sodium ion channels. So as that voltage will start to increase, that will cause more sodium ion channels to open. Then we have this positive 35 to 40 millivolts. What then happens though is we see this decrease, and that's because when it does reach this particular voltage, it then causes the voltage-gated sodium ion channels to close. So no more positive ions are entering, but we still have those potassium ions moving out. So we start to get this decrease, which we call the repolarization. That will then cause more potassium ion channels to open, so even more potassium is diffusing out. And eventually, so many positive ions have diffused out, we get to that resting state, but actually it overshoots and goes to minus 80 millivolts, which we can see here. We're now back to that original state of potassium ions would be able to move out, but all of the other gates are closed. So that is the generation of an action potential. And this happens along the axon multiple times. This graph is not a change over distance. So this isn't happening just once along the axon. It happens right at the start of the axon, and then at every node of Ranvier, if it is a myelinated neuron, you'll then have another action potential being generated until you get to the end of that neuron and then a synapse. So it's a bit like the idea of a Mexican wave moving along. If it was unmyelinated, then same idea, but it has to happen at every single position on the axon because you wouldn't have the myelin sheaths, so that would mean it would take longer for all of those action potentials to finally reach the end. Now you need to know about this all or nothing principle, and it links to the threshold. So if the axon isn't depolarized to at least minus 55 millivolts, which is the threshold to cause the rest of the action potential to be generated, then the impulse will not be produced. So what we mean by the nothing part of the principle is, if the stimulus isn't large enough to cause a voltage increase to at least minus 55 millivolts, then nothing more will happen. And that's what this bit is showing you here, some failed initiations of an action potential. The all part of the principle is the fact that if you do exceed that threshold, then you will always have an action potential, and it will always peak at exactly the same point. So even if the stimulus is bigger, so brighter light compared to dimmer lights, you will still have the same peak. But what changes is the frequency of those action potentials being generated. So that is the all or nothing principle, and the reason it's important is to make sure that animals are only responding to stimuli which are large enough rather than responding to every single slight change in the environment. Because if we did that, it would actually overwhelm your senses and it wouldn't necessarily be protecting you from stimuli which might be causing you danger. Now, the refractory period we mentioned is when it goes into this minus 80 millivolts. But the importance of this is, once we have this stage, we have a period when another action potential can't be generated straight away. And that's because these sodium ion channels, we describe as they're recovering. But essentially what we mean is, because we've now gone even more negative, you would have to have even more sodium ions to be able to reach that threshold. The importance of this is, first of all, it ensures that discrete impulses are produced. So an action potential cannot be generated immediately after another, and that makes sure each one is separate. So that helps with you being able to identify the source of the particular stimulus. It also makes sure that the action potentials will only be travelling in one direction along the axon. So it stops the action potential from spreading out in both directions, which would actually result in the impulse not travelling along. It limits the number of impulses as well, and this is important to prevent you overreacting to a particular stimulus or over-responding. Now, the factors that will affect the speed of conduction are myelination and saltatory conduction, the axon diameter, and temperature. So the first one was myelination, and that results in saltatory conduction. And what that term means is the action potentials will jump from node to node, meaning the node of Ranvier. And that is because this myelin sheath created by the Schwann cells is insulating, so the ions cannot diffuse in and out during these sections, so it jumps to the parts where there is no insulation. Now, as a result, that means that the impulse will travel much faster along the axon because you don't have to generate as many action potentials. You're only generating them at those nodes. Next then, the axon diameter. The wider the diameter, that is going to increase the speed. And the reason for that is basically like the idea of physics and wires and electricity. So the wider the diameter, that means we're going to have less leakage of the ions, and therefore the action potential can move faster. Temperature, there's two reasons why this has an effect. Number one, because if you have a higher temperature, the ions will have more kinetic energy. That means they'll be able to diffuse faster. The second reason is the enzymes involved in respiration to provide ATP for the sodium-potassium pump will be able to work faster as well if there is a higher temperature. Now, if it does exceed the optimum of around 37, then that could actually decrease the speed. So synapses are the gaps that we have between the neurons. So once you have your action potential reaching the end of a neuron, it has to get to the next one. And in order to do that, we have to have neurotransmitters, which are chemicals, diffusing across the gap, which will then generate the action potential in the next neuron. Now, the way that this happens is, first of all, your action potential, as I said, it will arrive at the end of the neuron, and we describe that as the synaptic knob. Depolarization of the synaptic knob leads to calcium ion channels opening, and therefore calcium ions will diffuse into the synaptic knob. Those calcium ions will cause the vesicles that contain the neurotransmitter to move towards the membrane, fuse, and release the neurotransmitter into the synaptic cleft. Because we have a high concentration of the neurotransmitter where they've just been released, they'll then diffuse across to the postsynaptic membrane. And on the postsynaptic membrane, there are receptors, complementary in shape, for those neurotransmitters to bind to. When those neurotransmitters bind to the receptors, it causes the sodium ion channels on the postsynaptic membrane to open. Therefore, sodium ions will diffuse in, and if enough sodium ions diffuse in to exceed that minus 55 millivolt threshold, then the action potential is generated. So that will then cause that action potential to be generated in the postsynaptic membrane, and then it can continue along the axon of the next neuron. Now, this is unidirectional, and you could be asked, how is it ensured that it's unidirectional? And there's a couple of ideas. Number one, the vesicles containing those neurotransmitters are only in the presynaptic neuron, so they can only be released from this side. Because of that concentration gradient, diffusion happens from the pre to the postsynaptic neuron, and you only find the receptors complementary in shape to those neurotransmitters on the membrane of the postsynaptic neuron. Now, one example of a synapse that you need to know is the cholinergic synapse. So it's exactly the process that we just went through, but you need to know the name of the neurotransmitter. So the neurotransmitter is acetylcholine. It will bind to its receptor, and then it will go through the whole stages that we said. But those neurotransmitters don't remain permanently bound. If they did, that would mean the sodium ion channels would be continually open, and you'd continually be triggering this action potential and a response, even though the stimulus wasn't present anymore. So to prevent that happening, there is an enzyme that will break down the acetylcholine into choline and acetate, which can then be reabsorbed into the presynaptic neuron and be reused. And that neuron is acetylcholine esterase. Now, I said that in order for the action potential to be generated in the postsynaptic neuron, you needed to have enough sodium ion channels opening, and that is caused by the neurotransmitters binding to the receptor. And there's two different mechanisms of summation to ensure that happens. Summation is referring to the fact that you have this rapid buildup of neurotransmitter to generate the action potential, and you can either have spatial or temporal. Spatial is when you have multiple neurons connecting to the same postsynaptic neuron. And collectively, these three neurons in this example should release enough neurotransmitter to cause enough of those sodium ion channels to open on the postsynaptic neuron. The alternative is temporal, and this is where you only have one presynaptic neuron connected to one postsynaptic neuron, but instead the way that this will cause enough neurotransmitter to be released is you have repeated releasing of that neurotransmitter over a short period of time, until eventually you have enough neurotransmitter bound to enough of the receptors on the postsynaptic neuron's membrane to open enough of those sodium ion channels. You also have inhibitory synapses, and in this case you would have chloride ions would be moving into the postsynaptic membrane. So you'd have different neurotransmitters binding to their complementary receptor, but this time it would cause chloride ion channels to open. And because you'd then have negative ions moving in, This would cause the membrane potential to decrease to at least minus 80 millivolts, and therefore the postsynaptic membrane would go into this hyperpolarization, making it very unlikely that an action potential could be generated. Neuromuscular junctions are what you have right at the end of your response arc. So you have your motor neuron, you then have a gap, and then you have your muscle. And your muscle is one of the effectors that would generate the response. And what you need to know is similarities and differences between the synapse that you find here at the neuromuscular junction compared to your typical synapse such as the cholinergic. So what they have in common is both of them will work in one direction only, so unidirectional. And that is because the receptors for the neurotransmitters are only found on the postsynaptic membrane, or in this case, the muscular membrane. A neuromuscular junction will only be excitatory, meaning it will only generate that response. Whereas a cholinergic, we looked at, could be excitatory, generating an action potential, or it could be inhibitory, which prevents the action potential from being generated. The neuromuscular junction will have a neuron connecting to a muscle, and it's always the motor neuron. The cholinergic is just a synapse between any two neurons. The neuromuscular junction is always at the end point for the responses, and the cholinergic synapse is always going to be generating a new action potential. For the neuromuscular junction, the acetylcholine will bind to receptors on muscle fibre, whereas the acetylcholine binds to receptors on the postsynaptic membrane in the cholinergic synapse. So that could be a long answer question, it could be a comparison table, so definitely learn those comparisons for those two. So that leads us on to muscles, which is an example of an effector, meaning it carries out the response. And muscles always occur in antagonistic pairs against the skeleton. And what that means is you have two different muscles that will be doing the opposite thing at the same time. So as one of them contracts, the other relaxes. Now this could be automatic or it could be conscious control for this movement. Now you need to know the structure of these muscles. So that's what we're looking at here. And we have the myofibrils labelled, and a myofibril is made up of lots of fused cells, and because they're fused together they'll share organelles. So they share the nucleus, they'll be sharing the cytoplasm, and this cytoplasm, because it's all shared, is known as sarcoplasm. So there'll be lots of nuclei within one of these myofibrils, but there's also a high number of mitochondria, and that is because we'll be needing ATP from respiration for the muscle contraction. Now these myofibrils are all bound together to make up this muscle fibre. But what we're actually going to focus on is the myofibril in more detail, having a look at the sarcomere. Now the muscle fibres are made up of millions of myofibrils, which collectively bring about that force to cause the movement of the skeleton. And the myofibrils are made up of two key proteins, myosin and actin. And that is what forms the sarcomere. The sarcomere you can actually look at in terms of different sections or bands, depending on whether you're looking at a part which is just made up of the actin protein, the myosin protein, or where the two proteins overlap. And that's what this diagram is representing. The actin is shown in red, the myosin is shown in blue, and what you can actually see from the myosin is the position. You have these chains of myosin with heads sticking off. That's what these lines at right angles are showing. But also the myosin molecules are all bound together, and that keeps them in a fixed position. The actin you just have as a thinner filament, and they can slide closer together towards this M line. Now you could be asked to describe what happens to the length of the different bands when a muscle contracts. So the A band is the length of the myosin, so that should never change. The A band is constant because the myosin itself, apart from the heads, the actual length of the myosin is never going to move. The H zone is where you just have myosin with no actin overlapping. Now that will change when a muscle contracts, because when a muscle contracts, the actin is sliding in, so therefore the H band or H zone is going to decrease because that is where you have myosin by itself. The I band is also going to change in size because the I band is where you have just actin by itself. So when the muscle is relaxed, you have a bigger I band. When it contracts, the I band decreases because the actin has slid closer together, so you have more of an overlap with the myosin. You could be asked about what happens to the Z lines, and the Z lines are indicating the start and the end of one sarcomere. And because the actin slides in, the Z lines become closer together as well. Now, my tip for learning this is, it isn't really intuitive, the letters representing these bands. So I'd say put them on a flashcard. So A band on one side, on the other side, just say it's the length of the myosin and it remains constant. The H zone is where you just have myosin by itself, decreases when muscles contract. So create a set of flashcards for this and just learn that content. This is just showing you the image as well of this happening under the microscope compared to the diagram. Now, the sliding filament theory is how the muscles actually contract. So when an action potential reaches the muscle at that neuromuscular junction, it's going to stimulate the muscles to contract. It does this by causing calcium ions to release from the sarcoplasm, and what this does is it causes the tropomyosin, which is the protein that covers the actin binding sites. It causes it to move out of the way, and therefore these binding sites are now exposed. Whilst ADP is attached to the myosin heads, which we can see here, the myosin head is able to bind to these exposed actin binding sites, and it forms what we call a cross bridge. The angle that this creates with the myosin head binding is going to result in some tension, and that causes this pulling or sliding action of the actin along the myosin. And we call that the power stroke, which we can see just here, when that head causes the actin to slide along. Now, in that power stroke action, the ADP and PI are released. ATP is the molecule that then binds to the myosin head and causes it to change shape slightly, and as a result, it detaches from the actin molecule. Within the sarcoplasm, there's an enzyme, ATPase, and that is activated by the calcium ions that were released in the sarcoplasm. So as long as you still have the stimulus causing these calcium ions to be released, then the next step will happen to trigger even more contraction. So that enzyme ATPase is going to hydrolyze the ATP that was attached to the head into ADP and PI. And that now releases energy for the myosin head to return back to its original position. And then we're back to the start again. That process repeats as long as the calcium ions remain high, the tropomyosin is moved out the way, and the myosin head combined. So this will repeatedly happen to slide the actin as close together so you have this full muscle contraction. So that continues to happen while the stimulus is causing that release of calcium ions. So you need to have ATP for this process to happen, but also a molecule called phosphocreatine. Now, if the muscles have been very active, we just saw there, it requires ATP. So the muscles require a high concentration of ATP to be able to contract. And the chemical phosphocreatine, which is stored in muscles, assists this process because it provides the phosphate. So in order to regenerate ATP, you need that inorganic phosphate to bind with ADP, and that's provided by the phosphocreatine. The last thing you need to know about muscles is two different types of muscles, slow twitch and fast twitch fibers, and you need to be able to compare their structure, location and general properties. So the slow twitch contain a large store of myoglobin, which is a type of hemoglobin which has a very high affinity for oxygen, and therefore it can hold on and store oxygen until the partial pressure is really low, and then it unloads. It also has a rich supply of blood, and that is so it can constantly provide oxygen, and many mitochondria, so you can have lots of aerobic respiration. So all of those structures link to the fact that these slow twitch fibres do contract slower, but they respire aerobically and for longer periods of time because of those structural adaptations. And that is for exercises which are going to be endurance, or it could be just regular exercises as well, just regular movements, nothing intensive. So for example, calf muscles when you're walking. Fast twitch fibre muscles are much thicker and there's more myosin filaments. They have a large glycogen store and a phosphocreatine store to help make the ATP. They also have a high concentration of enzymes needed for anaerobic respiration. Now, all of those structural features link to the properties of it contracts faster, short bursts of powerful contractions as well. So this is for intense exercise, and as we said, it'd be anaerobic respiration. And an example would be your biceps when you are contracting your biceps. So the last bit in this topic is homeostasis. And this involves physiological control systems to maintain your internal environment within set restricted limits. And the examples that you need to know are maintaining the core body temperature and maintaining the blood pH as well in relation to enzyme activity. Now, for those two, you don't really need to know much of the details. It's just knowing that those two conditions are controlled, and you'd need to know why in terms of the effect it has on enzyme activity. Homeostasis involves negative feedback, and this is when any deviation from the normal values, so if it goes too high or too low, there are mechanisms put in place to bring the conditions back to their original level. And it involves the nervous system and often hormones as well. Two key examples you need to know are blood glucose concentration and water potential. So we'll start with the blood glucose. Now, your blood glucose concentrations will increase after you have eaten food containing carbohydrates or drinks containing carbohydrates, and they'll decrease if you haven't eaten food containing carbohydrate for a while or if you have been exercising. The pancreas is the organ that detects these changes in blood glucose levels, and within the pancreas there are cells called the islets of Langerhans, which are responsible for releasing the insulin and the glucagon, which are the two hormones that bring blood glucose levels back to normal. Insulin is going to be released when blood glucose levels are too high, and that will bring blood glucose levels down to normal. Glucagon is released when the blood glucose levels are too low. So think of glucose gone, glucagon. And this will cause an increase in the blood glucose levels. Adrenaline, which is released by the adrenal glands, which are found just above the kidneys, this hormone is released when your body anticipates danger, and this results in more glucose being released from the hydrolysis of glycogen in the liver. Now, just to look at this as an overview, so your normal blood glucose levels, after you've eaten, they will increase. And this will be detected by the beta cells in the islets of Langerhans in the pancreas. The beta cells will release insulin. That will cause the liver cells to become more permeable to glucose, and there'll be enzymes activated to convert the glucose that's been absorbed into the liver cells into glycogen. That glucose then has now been removed from the blood and stored as insoluble glycogen, and therefore the blood glucose levels have been restored to normal. Now, if you haven't eaten food containing carbohydrates or you've done a lot of exercise, your blood glucose concentration will decrease. This is detected by the alpha cells in the islets of Langerhans in the pancreas. And it will cause the alpha cells to release glucagon, and also the adrenal glands will be releasing adrenaline. The second messenger model occurs to activate enzymes to hydrolyze the glycogen into glucose. That glucose is released back into the blood, and it brings those blood glucose levels back to the normal range. So we're going to have a look at some of those elements in more detail. So let's have a look at the action of insulin. First of all, it's the beta cells in the islets of Langerhans that detect when blood glucose levels are too high outside of the normal limits. And this then results in the beta cells secreting insulin in response to this. And these are the three key effects that insulin will have to decrease the amount of glucose in the blood. First of all, when that insulin binds to receptors on the surface of target cells, so on the cell surface membrane of the cells in the liver and also in the muscles, it causes changes to the tertiary structure of the channel proteins, which are the channel proteins responsible for transporting glucose. And this change in structure of them results in more glucose being absorbed from the blood into the cells by facilitated diffusion. So that's one way that the blood glucose levels will decrease. Not only that, but when the insulin binds to those receptors on the cell surface membrane of the liver cells, it causes more protein channels to be incorporated into the cell surface membrane. So both points 1 and 2 result in more glucose being absorbed from the blood into the cells. The final step is converting that glucose that is now in the cells instead of the blood into glycogen. And this step has to happen because if we don't convert glucose to glycogen, glucose is soluble, so it will lower the water potential of the cytoplasm in the cells, resulting in more water entering the cell by osmosis, and the cell might burst, or we might have lysis. So this conversion of glucose to insoluble glycogen is glycogenesis, and when the insulin binds, it also causes that to happen in the liver cells. So this is just a visual representation of some of those stages that we talked about. So we've got here the insulin hormone is bound to the complementary shaped insulin receptor on the cell surface membranes of liver cells. When it binds, what happens is actually a cascade of reactions that you don't need to know about, but it causes this release of chemicals, and those chemicals cause vesicles that are within the liver cells, which is what we're looking at here, these vesicles, which is just a sphere of phospholipid, and we've got the channel proteins embedded within that. And when these chemicals are released, it causes these vesicles, which contain those glucose protein channels, to move towards the cell surface membrane, and they then with that cell surface membrane. And as a result, you would then have all of these extra protein channels embedded within the cell surface membrane, so there's a larger surface area for that facilitated diffusion of glucose from the blood into the liver cell. The action of glucagon, again, there's three different actions. It will attach to the receptors on the target cells, and that would be the liver cells or the muscle cells. And when it binds, it causes the protein to be activated into adenylate cyclase. And this causes ATP to be converted into cyclic or cyclic AMP. That activates another enzyme, protein kinase, and that can then hydrolyze glycogen into glucose. So that is your second messenger model, how you have this two stages of activation of enzymes until we eventually get the hydrolysis of glycogen. It will also activate other enzymes to convert glycerol from lipids and amino acids from proteins into glucose. So this is the second messenger model. The glucagon binds to the glucagon receptors on the target cell, so for example the liver. That, once it is bound, causes this protein to change shape. And that change in shape to the enzyme adenyl cyclase or adenylate cyclase makes it activated. And the activated form can catalyze the reaction of converting ATP into cyclic AMP. And cyclic or cyclic AMP is the second messenger. So the cyclic AMP, which is this second messenger, is triggering the final stage to happen. It can convert the inactive protein kinase into an active form, and that is the enzyme which can then hydrolyze glycogen into glucose. Now this second messenger model happens for glucagon binding, but also adrenaline. So it's exactly the same process when adrenaline binds. Now adrenaline is the hormone that is secreted from the adrenal glands in response to anxiety, fear, so it's that flight or fight hormone. That's often how it's described. And the adrenaline will increase blood glucose levels. So if you did have to suddenly fight to survive or run away to survive, that would mean you'd need a lot of muscle contractions, which requires a lot of respiration, which requires glucose. So that's why adrenaline results in this increase in glucose, or that's why it's an advantage. The way it does that is, again, this second messenger model, much like we just saw for glucagon. So all of this is exactly the same as it was for the second messenger model for glucagon. It's just this time it would be adrenaline is the hormone that binds, and it binds to complementary shaped adrenaline receptors on the cell surface membrane of those liver cells, which are the target cells. But the cascades of responses resulting in the second messenger model is the same. The liver is involved in all three of these processes. Glycogenesis is when glucose is converted to glycogen, and this occurs in the liver and is catalyzed by the enzymes there. Glycogenolysis or glycogenolysis, pronouncing it like that so it's easier to remember, the lysis of glycogen is the splitting or hydrolysis. And again, this happens in the liver, and this is due to the enzymes involved in the second messenger model. Gluconeogenesis is creating glucose from other molecules, so the amino acids and glycerol, and again this happens in the liver due to the enzymes found there. Now, diabetes is when the body cannot produce enough insulin or it's not responding to insulin anymore. Type 1 diabetes is when people are unable to produce insulin or not producing enough. It normally starts in childhood and as a result of an autoimmune disease where the beta cells are attacked. So the treatment would be injections of insulin to counteract this. Type 2 diabetes, though, is when the receptors on the target cells lose their responsiveness to insulin. It usually develops in adults as a result of obesity and poor diet, and it's normally therefore controlled by regulating the intake of carbohydrates in your diet, increasing exercise, and sometimes it still involves insulin injections also. Osmoregulation is how the water potential of the blood is controlled. Blood with too low a water potential would be described as hypertonic. Blood with too high a water potential would be hypotonic. And if you had hypertonic blood, that means that too much water will leave the surrounding cells and move into the blood by osmosis, and that would cause your cells to shrivel or crenate. If you had hypertonic blood, that means that the excess water from the blood would move into the cells by osmosis, and it could actually cause the cells to burst. Now, neither of these are good, which is why we have to regulate the water potential. What could cause this is if you had hypertonic blood, that could be from excess sweating, not drinking enough, having a diet that's containing lots of ions, so salty diets. If you had hypotonic blood, then maybe you've drunk too much water or you don't have enough salt in your diet. So the corrective mechanisms for both. If you have hypertonic blood, more water has to be reabsorbed by osmosis into the blood from the tubules in the nephron, which is in the kidney. Therefore, you'll have urine which is lower in volume and more concentrated. If you have too much water in your blood, so hypertonic, that means less water will be reabsorbed by osmosis into the blood from the tubules in the nephrons in the kidneys. And as a result, you'll be producing larger volumes of urine, which are more dilute. Now, all of this happens in the nephron, and you need to know the structure of the nephron, which you find in the kidneys. The nephrons are these long tubules. They're surrounded by capillaries, and you have around 1 million of these in both of your kidneys. It's where the blood is filtered to remove the waste, but also selective reabsorption happens to reabsorb any useful substances back into the blood. So the way this happens is step 1, as the blood is going to flow into the glomerulus, the capillaries there, because there's a really, really high hydrostatic pressure, water and small molecules are forced out of the capillaries and into the renal capsule, which is this yellow part here, and the glomerulus is all of the red bit there where you have the capillaries. You've then created this filtrate, and that filtrate will then start to pass through to the proximal convoluted tubule. And this is where selective reabsorption occurs. So glucose is reabsorbed in the proximal convoluted tubule. The loop of Henle is where we maintain a sodium ion gradient, and that is to make sure that water can then be reabsorbed back into the blood. So at this point, the filtrate is going to be losing lots of the ions in the ascending limb, and that will then lower the water potential of the surrounding area around the loop of Henle, and then water will be able to move out by osmosis as it goes down the descending limb. So as the filtrate reaches the distal convoluted tubule, it's lost a lot of water, but also quite a lot of ions. And because it's lost a lot of ions, it is actually quite dilute by this stage. So more water will move out by osmosis, and at the collecting duct, even more water can move out to go back into the blood. The collecting duct carries the remaining liquid away to form the urine. Now that is a very basic overview. To have the full details, you will need to watch my full video where I actually go through every one of those steps in a lot more detail. This is just the basic summary video. That would not be enough detail though, so do check that one out up here. So how then this is regulated is using the hypothalamus and the posterior pituitary gland in the brain. The hypothalamus is where the changes in water potential are detected by receptors called osmoreceptors. If the water potential of the blood is too low, water leaves the osmoreceptor cells by osmosis, and that causes them to shrivel. And this stimulates the hypothalamus to produce more of a hormone called ADH, or anti-diuretic hormone. If the water potential of the blood is too high, water enters the osmoreceptors by osmosis. This stimulates the hypothalamus to produce less anti-diuretic hormone. So the hypothalamus is where the ADH is produced, but it then moves to the posterior pituitary, and from here it is released into the capillaries and into the blood. ADH travels through the blood and to its target organ, which is the kidney. Now what that hormone does is it causes an increase in the permeability of the walls of the collecting duct and the distal convoluted tubule, so they're more permeable to water. And as a result, more water will move out of the nephron by osmosis, and it's reabsorbed into the blood. So you'll have more water returning to the blood and more concentrated urine. So if we have a look then at this overall view of the homeostasis, negative feedback. We've got our normal water potential of the blood. Now the water potential of the blood could increase, so maybe you've drunk a lot of water in a short space of time. And that increase in the water potential of the blood gets detected by the osmoreceptors, which are within the hypothalamus in the brain. The hypothalamus will then produce less ADH, and therefore the posterior pituitary gland will release that less ADH into the blood. That transports around in the blood until it binds to the target cells, which are the cells lining the distal convoluted tubule and the collecting duct walls, and they then become less permeable to water. That means less water is reabsorbed into the blood and more stays in the nephron, which is then within the collecting duct, which goes on towards to make urine. So it results in a larger volume of dilute urine. And as a result, the water potential of the blood returns back to its normal limit. Now, the opposite of that would be if the water potential of the blood decreases, meaning you don't have enough water in the blood. And that could be you've not drunk enough water, or maybe you've been sweating a lot. And this again is detected by the osmoreceptors in the hypothalamus. And this time the hypothalamus will produce more ADH in response, which is then released into the blood by the posterior pituitary gland. That will then transport in the blood, bind to the receptors on the cells lining the distal convoluted tubule and the collecting duct, and that makes the walls more permeable to water. Therefore, more water is reabsorbed back into the blood, and as a result, there are lower or smaller volumes of urine, and it's more concentrated urine, and this results in the water potential of the blood returning into that normal limit. So that's it, topic 6 in a nutshell. Remember, this was the overall summary, and if you do want to see the more detailed versions, then check out my topic 6 playlist and you can see every single video in more detail. I hope you found this video helpful, and if you have stayed to the end, oh my gosh, then you are in it for the long run. Good luck with your revision, and if you've liked this video, please give it a thumbs up.